Okay. Okay, so our next talk is going to take us more into biological data. And over the last decade, gene expression profiling has really moved over to being very often at single cell resolution, and there's been a sort of explosion of methods developed for single cell RNA sequencing data and other types of single cell data. And so it's great to have Luisa here from Leeds, who's going to give us a kind of whirlwind overview of single cell RNA-seq data analysis. So over to you, Luisa.
Thank you. Okay. Good morning, everyone. I'm very happy to be here in Manchester today. I am Luisa Cutillo, as Max was saying, and I work in the University of Leeds, where I'm a lecturer. And my main research field is in application in biomedical problems. So originally I'm a mathematician working in a department of statistics, and I am working currently in some projects with single-cell RNA-seq data. And today I'm not talking about my research, but I am talking about what we need to know when we want to work with this data, because in order to develop methodologies and to progress with this data, we need to understand them, and we need to understand also how to deal with them. So there's a lot of pre-processing to do. So this is how I will organize the talk. There's a little bit of introduction on NGS in general, then to single-cell RNA-seq, and then we'll step to the data analysis and then discussion and question if there's time. So, introduction to NGS. So first of all, I know that the audience is pretty heterogeneous, I guess. So I just wanted to know if you ever worked with NGS data, or you know what they are. Yeah, who doesn't? Okay, who doesn't know what they are? Oh, okay, so this. Don't be shouting, you do know. But anyway, so next generation sequencing is also referred to massively parallel sequencing. And this massively parallel is, let's say, is the key in the NGS. And it refers to a set of technologies that enables the sequencing of thousands to millions of DNA. Is it me? No, I don't know, someone is here. Anyway. It's on me, I guess. So DNA molecules simultaneously. So that's why parallel, also deep sequencing. So when the word sequencing, say you need to do something? Yeah? Okay, I do not have any audio. Great. So the other word that was in the NGS is the sequencing. So what are we sequencing? So we're trying to sequence the DNA. And I'm very glad of the previous talk, because clearly this isn't, okay. Yeah, I had to wait for the animation to come. So the DNA replicates itself using chunks of bases that forms the words. And the words are made with letters that are the nucleotides, A, C, T, G, and there are about 3 billion bases, as was previously said in this talk. And the reason why we want to sequence it is because we are all different, although we are all very similar, because about 99% of those bases are the same in every person, but that the rest have 1% that makes us unique, and it's very fascinating. I find it very fascinating, the fact that we're so similar but yet so different. And there's a lot of interest in doing this and doing this fast. So again, a little bit of background. Why is NGS so revolutionary? Well, it all started with the Human Genome Project, where about 100 people donated blood. this project started in 1990 with James Watson, that is one of the scientists that discovered the double helix of the DNA. And then what happened, he started working on this project in what it was called the National Institute of Health. And then because he started having some issues about patenting genes, so anyway, he resigned. And he gave the role to Francis Collins in 1993. He took the lead, and then the institute was called National Human Genome Institute of Research, and it's still named like that, and they still look after, they still curate the reference genome. So the project was declared concluded in the 2003, where about 90% of the whole human genome was sequenced with high accuracy. And then it took till 2022 to complete the sequence of a human genome. And in practice, the bit that was hard to finish, it was completed the Y chromosome, that was pretty challenging. So there was this preprint going around about in December 2022, this is practically yesterday. So this is just to tell you how difficult this whole process has been. It took 32 years to have this reference genome. So that's where the revolution comes in. The NGS now takes only 1 day. So if you compare it to 32 years, it's pretty, let's say, an improvement. And so this can make you think of beautiful things that you can do. You can work on gene therapy, and you can sequence unborn children or children as soon as they're born, and you can do fantastic things potentially with this technique. So that's the power. So, but why is NGS so much faster? It is faster because it can sequence billions, it can let you sequence billions of strands of sequence simultaneously. So why didn't the people working on the Human Genome Project did so then? Because they didn't have the technology, they only could use something that is called Sanger sequencing, and it only allowed one strand at a time. So NGS, though, can work thanks to that project because they still created this reference DNA, bearing in mind that this doesn't refer to one person, it's a pool of the information from all the people that donated the sample to the project. So that's like a mosaic, let's say, and it keeps getting updated, so there's always new releases of the reference genome. So let's step to a typical RNA-seq experiment. So you can sequence both RNA and DNA. You have just to convert RNA to cDNA first. RNA is extracted from some biological material you're interested in, and then this is converted to cDNA, and then this is chopped in parts. The way this is cut is with some enzymes, or there's some sounds, waves, there's certain frequencies that can cut the DNA in pieces, and then some adapters are attached to these little strands, fragments. At that point, if that material is not enough for your experiment, there's amplification by PCR, and finally this forms your library ready to be sequenced. So there's a machine. This library is inputted into this machine, usually an Illumina machine, for example, there, and then individual reads are extracted. So they gives you for each fragment these letters we were talking about. And then those are aligned against the reference genome. This isn't obvious, there would be a lot of overlaps, so this is just counted, okay? And so each reference gene, let's say, on the genome will have an attributed number, that is the count corresponding to how many letters were aligned against it. So we have now done this kind of general introduction to NGS in general, we can switch to single-cell RNA-seq. So there's a new word there, is called their single-cell. So why single-cell? What was before single-cell? Before single-cell, and actually still a lot used, is bulk RNA-seq. What are the main differences? The main difference is that in the bulk RNA-seq analysis, in the bulk analysis, you have a sample, and then there's the DNA is extracted, but the RNA is extracted, and there's no memory of which cell this material came from. So you have a kind of average expression measured at the end. While with single cell, the input of your experiment would be the cells, so you keep track of which cells the RNA came from, and that will give you as an output, each experiment practically will be the single cell, and you will have track of that. And then you can look into interesting questions such as heterogeneity and subpopulation expression variability in thousands of cells, while the cellular heterogeneity is completely masked in the bulk approach. Okay? So those are the main differences. So bulk RNA-seq measures the average expression level for each gene across a large population of input cells. It is very useful, and it's still very effective because it can be used to compare transcriptomics, samples of same tissues from different species, etc. But it gives you an average measure of the gene expression across all the cells. But it is insufficient for studying heterogeneous system because of this limitation. While with single cell, we can measure the distribution of expression levels for each gene across a population of cells. It can give you an idea of the dynamics of the gene expression patterns, because the gene expression will change from cells to cells. And you can imagine that you start asking yourself a lot of interesting questions, for example, looking at cell-specific changes in transcriptome, or you can think of the identification of cell types, because some cell types are still unknown, some are known, and you want to find out if in your samples there's exactly the cell type that you're looking for, etc. And so the big power of this is that it can reveal the heterogeneity within the population of cells. And of course, a challenge is that it is very high dimensional. So it's thousands of cells. When you have the bulk experiments, you do not have usually thousands of experiments, and the number of experiments are smaller than the number of cells actually that you end up sequencing. Okay, and this, I just wanted to show you an image that is so usually brought up because it was really fun when yeah? No, depends if you have a targeted experiment in which you're labeling the cells in advance, then yes, but otherwise no. Because that's where the exploration starts. You don't know the cells, but you are able to isolate them. So you don't know what they are, but you know that a cell. You don't know that. So you have your sample, you extract the cells. There's protocols to do that, and I'm not the expert on that side, but I know that such protocol exists, and no, you don't know it in advance usually. Okay, so I was saying the fruit salad is the best representation of the single cell experiment, because you know you're eating pieces of fruit, and you know that there's difference. You know, that's the question, do you know it in advance? No, but you know that those are different fruits. So in this case, imagine we know that there's different fruit, we know which is which, but with bulk, you have that all mixed together in a smoothie. So that's the best representation visually. That gives you an idea of the difference between single cell and bulk experiments. Okay, and there's this citation from this paper that I found relevant because it says single-cell RNA-seq could play a key role in personalized medicine by facilitating characterization of cells, pathways, and genes associated with human diseases such as cancer. And this is used a lot, as I was saying, for this kind of purposes, this technology. Okay, so again, a little summary of pro and cons. One limitation, one challenge, rather than a limitation of single-cell RNA-seq is that the data are much sparse and the noise is much higher than in bulk. But the positive is that there's many more cells than bulk RNA-seq samples. Another positive is that they can help facing much more questions. You can answer much more questions than what you can do in bulk RNA-seq. And a limitation is that there's a lot to do still because we were doing great with bulk. We had a lot of methodology developed, but we cannot just apply them to the single-cell data. Sometimes you cannot really apply them, and you have to adapt them. There's a lot of development. There's also good news because we like to develop methods. So there's a lot to do. So novel methods are required. So what can we investigate with single-cell RNA-seq? Most relevant applications include as I was saying, the study cellular heterogeneity, the discovery of novel cell populations, and predict the cell fate differentiation, because you might look at cells that are during the differentiation process and you want to see what's happening there. So you want to infer that process. And that is done by cell analysis, because you can see you have a double problem. You can look at the problem when you got your data. You can look at the problem from the cell point of view, or you can look at the problem from the genes point of view. So if you look at the problem from the genes point of view, then you can detect the cell type, specific differentially expressed genes. So imagine you identify the cell types, and then you look at the differentially expressed genes. And that makes you characterize in this way the genes that are specifically differentially expressed in those kind of cells. And again, you can understand cell-specific type regulatory networks. While again with bulk you will not do that. You have a gene regulatory network. Here you can have it cell-specific. Okay, other applications include the building cells atlas. For example, there's a big project, it's the Human Cell Atlas, that has the ambition to create a reference map of all human cells. And there's a link there in the slides I will leave here, so if you want to go and explore this project. Okay. So, more in detail, an overview of a typical single-cell RNA-seq experiment. So you have your sample collection, and then there's this phase of cell dissociation. Again, there's different protocols. So you have your individual cells. So keep in mind that we're using there's different colors in this image. And then from each cell, you can extract your RNA. And then you see, again, different colors and different quantities. Then that goes into a sequencing machine, and that gives you the reads. So you have the association with the nucleotides. So you have the little letters there. So 3 colors again. And at this point, you do the alignments. You have your reference genome, thanks to the big project that we were mentioning before. And we align it. So we count, really, every time there's a match. We add the 1 in practice. And this translates into a count matrix. So you have this matrix with numbers, where practically you end up with the rows that are genes, or your reference that you consider, let's call them genes, and the columns are the cells. So in practice, each cell is a different experiment. Yeah? Not yet. So, no, you know which, the cell type is not identified at the moment. We just know that that's an individual cell, there's no label. I am adding a color just to, in these images, it's not my image, there's a color just so that you can see it's a distinct cell, but we do not have yet the cell type. We will do it in downstream analysis. We will go through all the process, so step by step. The first step is we have to read the data. I have to say that's where I start looking at data, when I get the numbers, not before. I don't run experiments. Okay, so we got this count matrix, and then it's a lot of information. You can't really look at the big matrix. So first thing that you can do to visually understand what's going on, if there's any natural partition of these experiments or actually cells, well, you can run a PCA, for example. You look at the first two dimensions and you, ideally, if that was the truth, the three different kinds of cells, but we don't know, then you would see them grouped there, but this is very ideal. Okay, and after this, you start running all the computational tools available to do your, to investigate the biological questions. Okay, so there's different yeah. Yeah, you have to do normalization. So you can't really start from this. You have to look at the data and check for any kind of issues that is affecting your data, and then you can progress towards the analysis. But I have all the steps for you because this is a tutorial, so we'll go through all the possible steps, and I gave you, and I prepared some links and some R tools, I mean, just names, we're not rounding them, so that you can try it yourself. Okay, so there's different platforms and there's different protocols, but what we are more interested in is the data analysis space. That's where I can be more helpful, let's say. So this is just to give you an idea of how these technologies are evolving. This is a snapshot given in 2018 by Benson et al. Where there's a different technologies on top. You see there's a single well, then the multiplexing, there's different wells, and then there's integrated fluid circuits, and then there's liquid handling robotics, the nano droplets, the big wells, and the in situ barcoding. Now, there's another one I'll show you after, because that's up to 2017. And then you can see that that's how it progressed in time. And then at the moment, with this droplets technology, we had an improvement so that we can have tens of thousands of cells that can be sequenced together. Okay, but more recently, there's this other one, this other work, 2022, that gives a snapshot including the spatial transcriptomics. So you can see again, we start from 1 cell at a time, 100 cells at a time, then 10K, 100K, 1000K, and more than 1000K cells at a time. This is massive. It's an incredible number if you think of it. And not only we have that, but look at the number of publications related. So look at what's happening. 2020 onwards, we have an enormous number of. A number is a beautiful word, I think. It's an enormous number of publications. That means there's a lot of research in single-cell biology. And the number of cells that we can sequence nowadays is massive. And at the top is very important also, the cost of the experiments has gone down a lot. That also helps the advancement of the research in this direction. So everything is becoming more possible and sustainable. Still, I think it can be very expensive, but it's more possible. Okay, so we have been talking about reads. It is fair to tell you that it's not the only things, this count number associated, actually is not the only things. Another thing that is pretty popular is the UMIs, is the unique molecular identifiers. So why this has been introduced? Because that PCR amplification phase, where your biological material needs to be boosted a little bit so that the experiment can run better, introduced nonlinear bias. So these UMIs is also called molecular barcoding, are short DNA molecules which are ligated to your fragments of DNA. So the way those are used to tag uniquely the molecule in the sequencing library before the PCR. Okay. So there's a certain number of possible UMIs, and L is the length of the UMI, and if L is too low, it can cause some problems. So the downstream analysis really should use only the sum of the distinct UMIs. And this is, let's say, in this plot here, you can understand why. So you can imagine you have 3 fragments here that are tagged to the UMI before the amplification. When there's the amplification with the PCR, you can see that there's clones in practice of the same sequences. So when you go and align, you can end up reading more than was actually there. So if you go back to the UMIs, you can see that originally you actually had 3 unique input fragments. Okay, so that's important. Okay. So you remove that kind of bias, amplification bias with the UMIs. Okay, so what is the difference between UMIs and read counts? As I was saying, the reads are the counts of all the reads that map to a given gene. UMIs is the number of distinct UMIs, and so it's the number of the original fragments that were sampled before the amplification with the PCR. Okay. Then after the PCR enrichment, if you will not use the UMIs, you cannot distinguish between the multiple copies caused by PCR, clones, or by the real biological duplicates. That is actually a real abundance of that fragment. So if you use the UMIs, the PCR clones can be found just searching for the non-unique fragment UMI combinations, and that can only be explained by the fact that there were PCR clones. Yeah. After, I'll show you after. Now we are just saying, oh, we want the numbers, which is the best number. So we're suggesting to use the UMIs where possible. Okay, then again, we look at the read counts here. So you see, when you use read counts, you have larger counts, includes potential PCR artifacts, you have large variability, and this happens both for full lengths and tag-based 3' to 5'. This is a protocol of how to do the reads, but so that you know. And then for the UMIs, you have smaller counts, you reduce the Poisson variability, and then there's no artifacts, and this is only tag-based, so it has specific position. Anyway, that's a technicality. And so regardless the count type, in both cases, you do have to face sparsity. So those data anyway would be extremely sparse. That means there would be a high proportion of zeros in what you get in the end. And these zeros is called zero inflation, and can be due to both biological reasons or technical reasons. And we will look at this also in this talk. Oh, sorry, the animation didn't go perfect there. Anyway, so issues with the single-cell RNA-seq system, just to mention, so there's transcriptional bursting. So we assume that the The process is a continuous process, but there could be dispersed. So the transcription can happen in bursts, and this can affect what you're measuring. That's the dropouts. That is a special kind of missing data. There's this amplification bias. There's also a problem when I show you all the wells. It can be that some of the wells are empty. There could be more than one cell in a well, so it's doublets. You can be measuring cells that are not good because they're dying, the membrane is damaged, so that introduces bias. Also, you have hidden factors that you maybe are not interested in analyzing, like cell cycle, cell sides. The cells do not have the same sides. You can imagine that affects the quantity, the biological material that you are examining. And so there's other things that are going on that we might not want to embed in our analysis. And also, there's strong batch effects and background noise that require to be adjusted. That was the question about normalization. So this batch effect is whatever is experimentally related, okay? And we will go through this. So single cell are much more noisy, but and also very sparse, but you have much more cells. So ideally, that will, let's say, top up for these problems, because when you have a lot of things, you can do more analysis. Okay, so we have done with the introduction. We can finally go to what you're all waiting for, that is the data analysis. Okay, so data analysis. Those are some reference papers and tutorials. Again, I'll share the slides so you have them there. So my favorite is orchestrating single cells analysis. It's in bioconductor. Bioconductor is an extremely good resource for tools. It's always up to date, and so these orchestrating single cells always up to date also. Those are other tutorials and papers that I found relevant for you to have. So in your own time, if you can go and just play around with these ones, I think it would be very helpful if you want to start or continue your study in single cell. So that's the set of pipelines that I thought I'd bring to you. So this is an ideal approach. You start from the read alignment and the count matrix. Then you have quality control and filtering. You do your normalization and batch effect removal, and I'll explain all of this, what we mean with these words. Then there's feature selection and dimensionality reduction, because you've got enormous amount of data, and you want to keep just relevant ones for your analysis. And then there's all the downstream analysis that will take you to the interpretation. That means clustering, trajectory, inference, differential expression, and finally the interpretation. That way you will need to work in a multidisciplinary team for that. Actually, I suggest from the beginning, because there's a lot of choices that you have to make, and you don't want to do something that looks reasonable based on the numbers. You also want a good interpretation, if possible, from the beginning. Okay, so that's the overall number of steps. Those are the steps that we should perform when doing a full analysis of single cell RNA-seq data. So I feel like we covered the first one, that is the counts. So these pipelines will depend on the system, the row count types, and also the downstream analysis of interest, because maybe you're interested in looking at the differential expression in genes, or maybe you're just interested in clustering, or maybe you're just interested in trajectory estimation. You might not want to do everything, okay? Also, you might be interested in identifying specific factors that are called confounders in the data set, etc. So you can omit some steps. Some are essential, some others you do not have to run. But I suggest you always start from visualization and data exploration. That's essential, because let's say You can look at the data with a macroscopic approach. Let's say you zoom out, let's say, and you can understand if there's anything really terribly wrong in your data. And that's essential, because from the numbers in a file, you really cannot do that. Okay, so there could be outliers, for example. Okay, so this is all associated to a lot of computational challenges, of course, because there's still the lack of golden standards. Because even if we did done so much advancement, the field, I think, is still relatively immature. Sorry, the number of tools is not 400, it's 1,526, or I don't know why it's written 400 there, but it's 1,526 as the plot says here. So currently, that's the number of tools we got. So there's an enormous choice in a lot of categories. So depending on which analysis, you can read them. They're in the graph that I have there, but if you click on if you go on this scRNA tools website, they're all there. And again, there's a very good link for you. If you go there, you can see this image, and also you can access all the tools. That's very good. Okay, so there's an enormous amount of choices. When we have to make choices, we're likely to make mistakes if we don't understand what we're doing. So be careful when you choose what to do. These pipelines are really complex. Sometimes you have to mix a lot of steps, and sometimes in different programming languages still. Yeah, it's another challenge. What you end up doing, it really depends, again, on your question. So have clear in mind what you want to get out of your data. And also the data size, the dataset size are enormous, and this is challenging. You have to have access to cloud computing or clusters, and there's this zero problems. How to interpret the zero, how to get rid of the zero, shall we get rid of the zero, and then we will talk about later. So we say that we saw more or less how to get to the numbers through this sequencing machine, but then we need to understand if the numbers that we get, first of all, are good enough. So we have to inspect the sequence quality. FastQC is one of these projects that look after that. Again, there's a link. So FastQC helps you remove reads with low quality. Those are tools, Trim, Galore, or Cut Adapt. Then the problem of the multiple barcodes is called demultiplexing, what you do. So you can group reads by cell barcodes. And the alignment is done with tools like Star or pseudo-aligner Callisto or Salmon. You feel free to suggest more. To quantify reads versus transcript, there's a lot of tools widely used. It's called RSEM, HDSEC, or RSubread, or other specialized tools like Collapse UMIs and the multiplex. And if you're using UMIs, you have to collapse UMIs to count on genes and transcripts. So I'm saying genes, but you can have transcripts that are not necessarily genes, okay? So this is the part that relates to row counts. So there was the question of row counts. So you have, I don't want to say problems at the beginning, but you have to keep this in mind. If you're lucky enough, someone did this for you, and you get just the clean numbers. Yeah? Yeah. Yeah.
Realistic to use the transcripts, aware methods like RSM and ELLIS Summer. Do you think they're going to work worse on a single cell than
Yeah, Yeah, I think that is an issue, but do you have?
It's just,
Yeah, there's an expert here.
it's just someone in my group.
Okay. Yeah. Okay. No. No, I don't have that specifically. So, so the person is in your group is here. Sorry to put you in this on the spot. What are you doing for this bias on the 3 prime? Yeah, you have got a microphone. Yeah.
Yeah, I think even for the 3' attack sequencing, it's valuable to identify different isoform. But since the isoform will share the similar 3' UTR sometimes, but if you study like the alternative polydeniation, which means the different position is the poly tail, it could give more extra information, so it could be better to analyze like the clustering or something. And since the 3' attack only captures 3' end of the transcript, so I think it's valuable to study the APA, the alternative poly A.
Study what?
Alternative poly A site. Yes, because they only capture the 3' UTR and use this APA, the alternative poly A profile as estimator like the transcripts. I think maybe it's useful. And using the Simon kind of tools, you can generate the matrix, like use the isoform, like use the transcript information. So even some transcript could share the same 3' UTR. You can also identify the more transcript-like matrix instead of the gene matrix.
Yeah.
Yeah. So that's what I did.
So basically studying transcript level rather than gene level.
Yeah, I mean, yeah. I think, there was a nice paper from, what's his name? One of the Salmon authors, showing that even with bulk RNA-seq, quite a few transcripts are difficult to disambiguate with short read data. Like there's quite a lot of uncertainty about which transcripts you can pick out.
Yeah.
But I suspect with single cell it's even worse because the distribution of the reads is so much more 3' biased. But what Kian's been looking at is alternative 3' UTR usage and alternative polyadenylation sites. And there's a lot of power for detecting that in single cell data. Like that is something you can, so that can distinguish transcripts often. But it might be difficult to distinguish like all the different transcripts. That was just, yeah.
No, I
Sorry
think
to butt in.
No, actually, this very, no, honestly, this is, so I have to admit that I like to jump in after all this done. But I do know that this is an issue. And let's say, like when you put your hand in the sun, they say, I don't know, there was an issue. So, but if actually this is relevant, if you leave in these links for the tutorial, if you can just send me any reference to add here, and maybe we can actually add another bullet point mentioning this bias, because this is very, very important. So in practice, if you got bias at the beginning, you're kind of affecting all the analysis. So it's very relevant. It's okay. Perfect. Not perfect. Send. So quality control and filtering. Moving on. So you have quality control at gene level and quality control at cell transcript level and the cell level. Okay. So what you want to do, you want to remove potentially the doublets, empty drops, the one that empty drops and wells and writing missing cells, and then you want to remove the damaged ones, dying cells or broken membrane. So there's some metrics that helps you do that. And the same thing with the gene transcript level. For example, you want to remove genes with very high expression or genes that are not expressed, okay, because when dealing with dropouts. Okay, how do we proceed with this? So for example, this is an example. This is when you filter low quality cells. In practice, you will see that this is a very small example. There's some cells there, cell 3, that has got consistently lower expression than the others. And the same with the gene D in this other example. You see that that gene is consistently much, much lower than the other, like assuming that there's a noise threshold rather than a low expression in general. So these quality metrics can be computed on the row counts so that you can filter out problematic cells or genes, and you can look at the metric distribution before deciding on the cutoffs. You might want to consider joint rules to do that. So I suggest you visualize the impact of your quality controls cutoff. So you could plot the PCA components, that much more advanced techniques. I'm just saying the fastest, the easiest way to proceed. And then QC metrics to help identify outliers. And after you've done your choices, maybe go back and replot to see if that has been effective. Or if something more is needed or something less is needed. So you can do all of this with this package. It's called scatter. And there's a function called calculate QCM metrics. So this provides cell-level QC metrics and feature-level QC metrics. Your features, in this case, could be the genes or the transcripts, okay? So it's the two directions you can go. Also, scatter is really good because it gives you diagnostic plots, filtering, basic normalization, and other visualization plots. So it's good to explore this tool. Okay. And this is an example. So you can have, this is just you run the tool and you get histograms like this, where you have the total counts, histograms, total counts of cells, or you have the number of cells. Sorry, you can't read. It's called number of. There's a number of expressed genes at the bottom, and this is one of the plots that Scatter produces. So you can see the total counts. So it gives you different sizes based on the numbers, and then you got these 2 dimensions that you see the PCA. Okay, this is just an example. Now, spike-ins, we didn't touch on spike-ins. Shall we use spike-ins or not, or can we use spike-ins or not? So what are spike-ins? First of all, so you can use external molecules that can be added in your experiment with a known concentration. So those are molecules that, in principle, you should know what they're doing. And so spike-ins are usually used for quality control, because you know what should happen with a spike-in, and you go and check if that happened. You can have an idea of noise, the technical noise, or actually the dropout rates, your capture efficiency, or any amplification that was due to low starting amount of RNA in cells. Also, whatever is measured with spike-ins can be used for data normalization, because you can imagine if nothing should have been measured correspondence to the spike-ins, then you can normalize all your data based on that value. But there are problems, because spike-ins do behave different to endogenous genes. And also, when you plan your experiment, those are not easy to dose. They do require extra depth. And you cannot use them in droplet-based approach, for example. Okay. yeah, so you can just see those plus in that plot there are the spiking molecules. So you can have them in your experiment. So you see cell 1, cell 2, you can have those spikings, and you can see how the spikings behave in the two different cells, given that those are the same spikings. Okay. The other thing I wanted to mention are the dropouts. So what are dropouts? So the sparsity of single-cell RNA-seq is due to these dropouts. So zeros that arise in this kind of experiments are happening for different reasons. So if a gene, I call it gene, but again, it can be anything, was not expressed in the cell, then that means that there are not transcript to sequencing correspondence to that gene. So that is a true zero, that is the biological zero. And there's nothing you should worry about. But the problem is that you don't know what is a biological zero, because what can happen is that a gene was expressed, but for some reason, capturing efficiency, that what I was mentioning before, this bursting, this transcription bursting, the transcript was lost and you didn't manage to sequence it. So this is a proper dropout. Or the gene was expressed, the transcript was captured, but then the sequencing depth was not sufficient, and then you have a dropout again. So we have to be able to distinguish between these cases, and also if it is a dropout and it isn't a biological zero, you want to replace that zero with something. So there's a lot of literature that has been developed to do so, and it's called data imputation. So data imputation aims to replace the zero abundance values with these expected values with a model that is called dropout model. Now, imputation is arguable and difficult, and it creates the risk of introducing false positives. That means was it a biological zero and you actually imputed something, or did you impute too much, etc. So it can create false positive in downstream analysis. And in alternative, you can use some more implicit approaches that are called zero-inflated distributions. So those are some references for you there. There's this MAGIC, DR impute, SC impute, and then there's this latest paper about zero preserving the imputation of single-cell RNA-seq data. Okay, so we touched on this other topic here, and that's the references for you. Okay, that's where we are there. Normalization, finally, one of the questions. So what is normalization? Normalization aims to remove the non-biological variation. So because the variation that you observe is, that you want to observe, is the biological one, but there's a lot of other variation that is not biological. It's bias, called bias, and you also want to make the count distributions comparable, because you might imagine that depending on the setting of your experiment, on the size of the cell, etc., you might have these distributions that are really out of scale or not comparable at all. So you want to do both things, make the count distribution comparable and removing the systematic non-biological variation. So that's a reference paper again by Valleios et al. Normalizing single-cell sequencing data. This is a starting one. It's a very good one. The bias that we observe is due to low mRNA amount per cell, variable capture efficiency of this mRNA, variable sequencing depth among your experiments, and technical patches and others. So there's also other reasons. That's a note on normalization depends on the type of row counts. So 3' tagged or UMI counts. In this case, only cell-specific normalization is done. So between cell normalization, because you assume you do not have other kind of biases in this case. well, with the full-length read counts, the normalization for gene length you could also do, because the gene length would be more variable, so you do the within and between cell normalization. So bear in mind what kind of data you got before doing the normalization. How am I doing the time? So should be let's see. Okay. So normalization can have a very large impact on the downstream analysis. Main factors are there's global scaling or nonlinear account for library sites, sequencing depth. There could be batch affected and other confounders, can be known or unknown. So the package that I suggest you use, again, is SCONE. SCONE provides a wrapper to different normalization methods, and also there's 8 metrics to rank them on a data set. So also then there's SCRAN, and it computes the convolution-based scaling factors, SC norm, and the ST transform package. So those are all packages that are worth trying. So most of the genes, you have to assume that most of the genes are not differentially expressed. So there is no method that is good for all the data set. So it will really depend. What's best for your data set will depend on the data set. So again, you start from an exploratory analysis, and you also understand what kind of issue your data have and what kind of normalization you might need. Okay, so those are scaling approaches for normalization. Classic normalization methods for bulk might not work well for single-cell RNA data because of the zero inflation due to dropouts and transcriptional bursting. So the main approach are to adapt these size factors, but they might not work. So it's better to go to pooling and deconvolutional approaches. There's also regression-based approaches, and again, there's SC norm uses a two-step quantile regression, and I mentioned it before already. Okay, so there's a global scaling and nonlinear scaling kind of normalization. So, and this is the list of tools. Basically, these red ones are more complete tools that also include normalization, yeah, there are more general purposes ones. So those are the tools. I have the slides coming up a lot for remembering which tools. Okay, so we've done that. Batch effect. Batch effect is another very important thing to consider, because data might have been generated in multiple laboratories with different protocols, different times, etc. So you might notice differences in your data that are not actually the biological difference that you are after. So the factor that you see there are technical batch effects. And so the expression of the genes in one batch will be different from the other, but it's a systematic difference, not a biological difference. So this could mask, if you're not careful, the biology that is under, and introduce structure in the data that are not actually there. So that needs to be corrected. So if you look at the top plot, you can see that there's two different kind of things that you should observe, the triangles and the circles, just that they were done in different settings that are the colors. So what you observe is the left, but the ground truth is the right. So you have to reach that. And how do you do that? You have to apply methodologies that correct for this batch effect and unwanted variation. So this is an example of a regression model that models the big data set you're observing with biological factors, known unwanted factors, and hidden unwanted factors, because you might not know other factors that are still unwanted. You just know that you don't want them, but you don't know what they are, so you put them in the model. So you don't want to be too aggressive with your data, because you might end up filtering out the signal. Okay, so that you need a balance between wanted biological variation and removing the batch effect. Okay, those are modules in R. In Limma, you have remove batch effect, RoofSec, SVA-sec, combat, but also there's other approaches. Emerging approaches are mutual nearest neighbors, CCA, canonical correlation analysis, and also integrative non-negative matrix factorization in Liger package is called. Okay. So again, do not worry, I'll leave the slides, so do not rush right now. So it's fine. Okay. So this is an example when you do not apply batch correction and you do apply batch correction. So it's not totally clear, but you can see colors, but the colors are the different, it's not the biology, are the different batches. So clearly, when you visualize this data set with one of the tools we were mentioning before, you could see the batches. But when you do the correction and you redo the plot, you can see that all those colors are mixed, so all the, consider different labs are now not an effect anymore. It's not a factor anymore because things are integrated. Okay, so this is another example that you can use. So this is data integration. It's again like batch correction, because in practice you've got different maps of the human pancreas up there. So you have cell sec, then you have fluid diagram, you have in-drop, etc. And you have 8 different maps. So if you don't do not use any tool for data integration, when you use a tool that I will mention later, it's called Disney, on the left, you can see that those cells are clustering by batches, right? But when you do data integration, they're actually clustering by cell type, ideally. Well, this is a claim, but at least you don't see the effect of the batches anymore. You see clusters, but all the different sources are mixed together in the same cluster. Okay, so again, data visualization is very powerful. It can be misleading, but it's very powerful to highlight things like this. If, for example, you do data integration, did it work or not, you can just plot and see. This is this name, plot. Okay, and yeah, I left there's a tutorial on data integration on Seurat, and the link is there. Okay. Done this. Now, feature selection and dimension reduction. When we talk about features, again, we think of genes of transcript. We have very many of them. Are they all worth keeping into your analysis? So we would like maybe to select just a subset of the most biologically associated genes associated to what you want to observe, your biological problem. So, for example, you might want just to keep in your analysis the highly variable genes, the genes that are doing something. Okay, they're changing in your observations. And also, you might want to reduce the dimension, so you can have a linear or non-linear embeddings to project your multidimensional data set in a lower dimensional space where you can keep the biological signal, but everything is more handleable. You can actually work better. Obviously, you cannot visualize anything in more than 3 dimensions, but you can imagine that you can work more easily computationally on the lower dimension. Okay, so you have your expression matrix, you can have feature selection, and then dimensionality reduction. So you end up with a smaller data set that still keep all the information that you want. Hurry up a little bit, I feel like running out of time. So this is a way of keeping only the variable genes. This was an approach proposed by Brenneget and Doe in 2013. I still think it's very valid. There's other approaches, of course, but I still use this one. So the assumption is that the large differences in expression across cells are due to biological differences. So there is a positive relationship between the mean expression of the genes and the variance. So the high variable genes selection consists in identifying the genes that have a high cell-to-cell variation. So in practice, what you do, you normalize the data, you calculate the mean and the square coefficient of variation, and then you fit this quadratic model for the relationship between mean expression and the quadratic coefficient of variation that's defined there. And then you use a chi-square test to find the genes that are significantly above the curve. So in practice, this is a way of not using a cutoff, let's say, but you just do it smoothly, fitting the curve and doing a test. Let's say it's less brutal and more meaningful. So for the dimensionality reduction, we have many ways. We start from the most simple, that is the principal component analysis. It's well known. It's linear and interpretable, and is based on the singular value decomposition. That's a sketch of it. So we want to retain a relevant number of components, but this depends on our aim. We want to keep heterogeneity in data, so we hope that, for example, the first 2 dimensions will just keep all the variability of the data, and that's again the ideal situation, but there are plenty of other methods based on matrix factorization, so that's independent component analysis, NMF, or graph theory, for example, Disney, UMAP, ISOMAP, diffusion maps. There are also specific approaches for single cell that keep in mind the problem of the zeros. So there's the zero-inflated factor analysis, ZIFA, or the zero-inflated negative binomial bonding variation extraction that's called Zimbabwe. Let's say Zimbabwe, I don't know. Let's call it Zimbabwe. Sounds nicer. Okay, so those are the ways to go. Disney, the T-distributed stochastic neighbors embedding algorithm, is nonlinear. That means it's less interpretable. And it preserves local distances. That means distances that are between cells that are neighbor. So it provides a local embedding, so it doesn't preserve the global data structure. So when you look at the Disney plot, it doesn't always mean what it looks like. Okay? Locally, yes, not globally. It is iterative and stochastic. It depends on initialization seed. You will find out that if you change it, it keeps changing. And other tuning parameters. On large data set, it implements an efficient algorithm that is based on a fast Fourier transform, just to mention, that is also efficient on large data set. UMAP, this is also a very, very, very commonly used, uniform manifold approximation and projection. It's again non-linear and is based on topological structure, provides local and global embedding. So you look at it and kind of, okay, what you see is what it should be. And it's not completely stochastic, and that's got many tuning parameters, and it's fast. So let's see where you can find them. You want to use them, where you find them in the Surat 3, Scatter, Monopole 3, X, etc. So there's other packages, those are 3. So UMAP's becoming very popular. I use it. So again, just forget this again, some packages I wanted to remind to you. So just a visually comparison of the 3 methodologies I was telling you about, the PCA, Disney, UMAP. You can see that they should be doing the same thing, Do you see the same plot? So, no, I mean, more or less you can see that they have the grouping, same color of cells, but the outline is different. Okay. There are other visualizations, I was saying, diffusion maps. It might be appropriate when you're looking at cell differentiation. They're non-linear. They're used for dimensional reduction, and in particular when the distance between the points are measured as probability from stepping to one to another. So use diffusion maps when you are in those situations, because if you're looking at this problem of cell differentiation, clustering will not give you any idea of dynamics. Okay, getting to the end, downstream analysis. How long? Okay, so there's a lot of things that you can do at this level. So you can do clustering, then you can do annotation of the clustering, compositional analysis, trajectory, inference, cell markers, identification of the differential expression within clusters, between clusters, etc., gene regulatory networks at cell levels, etc. Okay, so you can have, again, these you can do at different levels. You can do cell level analysis and gene level analysis when you go downstream, because again, you can flip the problem. Okay, for the most popular clustering approaches are hierarchical clustering, k-means clustering, density-based clustering, mixture model clustering, or graph-based clustering, where the cells are first organized in a network using, for example, a KNN approach, and then each cluster is identified as a module of the network. What are the challenges when you do the clustering in single-cell RNA-seq? So first of all, what is the number of clusters? Some methodology might guess the optimal one, some others don't. What about dropouts and other technical issues? And then scalability. The number of cells could be millions, and some tools do not scale well in this case. So that's to be considered. And this is a table of clustering methods that is actually in the paper for DBScan. So there's many, as you can see. And those are some reference papers that talk about clustering. In particular, you see this methodology here, SC3. It does a PCA plus a k-means. It's very accurate, and it's highly complex and is not scalable though. So there's a lot of space for improvement, and again, scalability is the main issue. Surrogate clustering is the one that I wanted to focus on. Starts, as I was saying, with the PCA, construct the k-nearest neighbor graph in the PCA space, and then refine the edge weights between any two cells based on the overlap in the neighborhood. So, and the distance that is used to do so is something that's called Jacquard distance. That counts the shared X practically. So the way the cluster are there computed is computed in a way that optimizes a measure on the network that is called modularity. Practically, the algorithm is called the Louvain algorithm. It's in R, very simple. Just run iGraph and it's in R. And the advantage of this is that it doesn't require to choose the number of clusters. It optimizes the number of clusters. Okay, now imagine we got all done. How do we assess the clustering? So, is it a good clustering? Is there an interpretation? Is there a meaning? So we can, first of all, assess the structural validity of our clustering. We can visualize it. We can have an average silhouette. We can have a modularity score that tells us how well clustered the network is. You can apply, you can also compute this adjusted random index, and you mainly have to do biological interpretation at this point, because you can find out if your network and your clusters are good, but are they also meaningful? Are structurally good, but also meaningful. So part of my research addressed these questions, but I'm not talking about this now. So also you should check on quality metrics in your cluster, because you might have cluster with technical issues. And also, just an initial question, the cluster might not represent cell types. You wish that they did, but they might not, okay? And that's where, if you have any information to overlay, it would be relevant to validate your network. Okay. So, cluster annotation, cell identity. So you should try to make sense of the data. Samples are heterogeneous, either in composition and abundance. And then, so you can have manual approach, so you can have markers for the cell or for the genes to identify the population that you're looking at. You can use database that have cell type gene signatures, or you can have automatic approaches with mapping classification like SC-map single R. When you got tumor cells, this is much more challenging because there's much more variability. And also, when you identify this group, is that the subpopulation of cells? Is it functionally characterized by something? So there's a lot of investigation to do on your cluster once you find them in practice. And having an expert looking at the data with you is crucial for the interpretation. Okay, so you should not call them cell types when you identify them. You should say cell identities, because you find these clusters, but then you try to it's before, I think you can distinguish the names. But anyway, this is just an example. So you can try to annotate your clusters, and you do the best that you can, and more or less you can assume that the majority of the, so if cells are in the same cluster, belong to the same category, if the majority belongs to a specific type, then you have identified that type. But this again is an assumption. Okay, then there's another couple of things. Compositional analysis. So you can have compositional structure in a cluster. That means that there's proportion of cells that falls into cell identity cluster, and their change response to a disease treatment is also relevant. So you can investigate compositional changes in in single cell analysis data. But you need to have enough sufficient cell numbers, because you need to assess these cell identities, and sufficient sample numbers to evaluate the background variation in practice. So there's emerging methods. They come from mass cytometry and microbiome to do this kind of analysis. This is pretty recent. Trajectory inference, I think this is one of the last topics that we should talk about in this tutorial. So when you got your cells, the cellular diversity cannot be captured fully with this something that is static, so with a discrete system such as clustering. So the biological processes that we are saying are continuous process, but here you got snapshots of things, and so you got gaps in between, and you want to be able to identify the transition between cell identities. Okay, so you got your cluster, so you say you have your identities of cells. You want to understand how there's a transition between one and the other. So we need dynamic models in practice. So as I was saying, the trajectory will give you the opportunity of putting together all these snapshots of a continuous process, and you aim to find a path through the cellular space that minimizes the transcriptional changes between cells that are neighbors. Okay. The ordering of the cell is something a bit tricky, because what we think about ordering is not the biological ordering. So this ordering is called the pseudo-time, because the biological time is not exactly our time, so it's called pseudo-time. And that is actually a variable. So what's the connection between pseudo-time and branching? So pseudo-time is an ordered abstract unit of progress. So, and it was introduced in the original paper Monocle. Now there are Monocle 3 version, I guess. They're much better than Monocle now. And so you can imagine that if there's an evolution, there could be a kind of trajectory. There could be like cycles, linear, bifurcation, multiplication. Those are all, let's say, simplification of what can happen. So there can be a tree, connected graph, disconnected graph, etc. So there's many possibilities. In how do we do this inference? Which tools do we use? So again, the original paper and tool working on this problem was Monocle. Now there's Monocle 3. It's much more improved. The first one really didn't work greatly, but Monocle 3 is doing well. And then there's all the other ones. There's Ruder, Race ID, etc. And there's many others. They differ in accuracy, scalability, stability, etc. There's no best answer for this. So, and also when you start asking yourself questions, so are you sure that you have a developmental trajectory, for example? Where do you start? Where is the process starting? Where is it ending? And there's many methods available at the moment. And this is a reference paper, for example, from 2019. Okay. Differential expression is something that was very commonly used in bulk RNA-seq. And the methodologies that were available back then were HR, DSEC, etc. But single-cell RNA-seq data are much more sparse, higher variability, and many more cells than number of samples that we usually have in bulk RNA-seq. So single-cell specific methods are SCDE, or model-based analysis of single-cell transcriptomics. And the kind of approach that you do is you first identify the cell types, if it is unknown. Okay, you first do your clustering, and then you compare expression levels between populations. That's how you do. And so the kind of, what you want to do, you want then to identify marker genes within your subpopulation of cells, yeah? Yeah. Cells. Yeah. Yeah, I should have said identities, yeah. Cell identity. So the groups you identify in practice, so you did your best, and you end up with these groups of cells. And now you want to characterize them. So because you're still trying to understand what you got. And then you want to find marker genes. And so you look at differentially expressed genes. But yeah, that's a good point. Better say identities. Unless someone labeled the cells for you, and you can call them cell types. I mean, this happens in, there's some protocols to label cells, but then I think it's a different kind of experiment. So anyway, so you can compare genes within your cluster of cells, or so outside. So you differentially express genes to compare at least one cluster to another. You can do all sorts of things depending on your questions. Okay. So in Scrum, there's this very useful function. It's called find markers. So all your questions, Scrum will just answer for you. Just set the right input and run it. Okay. And gene regulatory networks. When you're comparing cell types, or as we want to say, identities, you want to find cell type specific networks. So you have this cluster of cells, and then you can find the networks of genes within each cluster, okay? And so you can compare the different cell populations that you have identified through the networks and understand the dynamics during development this way. Okay, so in practice, another way of comparing this cluster of cells, this population of cells that you identified, is through the gene networks. 2 minutes, we're done. I think we're done. I think was other applications. Identification of rare cell populations. I didn't touch on alternative splicing, allelic-specific expressions, and other kind of cell atlas, or spatial transcriptomics. I know that someone is working on spatial transcriptomics in this room. Another very interesting way to go is multi-omic single-cell integration. And there's a lot to do on the interpretation of the results. So you can do all the steps that we talked about, and then still there's a lot to do. Those are some reference papers, and those are the conclusions. So this is the last slide. So single-cell RNA-seq is a novel, a rapidly evolving technology. It lets us investigate a lot of questions, cell heterogeneity, dynamics, development, etc. And the level of resolution is much, much higher than the past. So we know more and more about, okay, with more and more accuracy. So the problem associated is that there's a lot of noise, and those data are highly sparse, and the interpretation of the zeros and how to deal with the zeros is a problem. The process to clean this data, to let's say pre-process the data, is general, but what you would choose at the beginning will affect your downstream analysis. So it's very interesting to see, if you have time, when you do some choices at the beginning, how do these affect your downstream analysis, and check if there's any best choice based on any criteria that you might think of. Then there's a lot of, as we were seeing at the beginning, above 1,500 meters, a bit more. So, and they're still not a golden standard. They're still, doing pick this or pick this other one. So there's no golden standard. We're not there yet. There's challenges in scalability because the data size increase so rapidly that the scalability doesn't keep up. So there's problems in terms of memory and runtime of a lot of methodology. Also, when we think, let's estimate the networks in the clusters that we identified, also there's a lot to do. And novel applications are continuously proposed. There's a lot of development going on. Have you seen the number of papers is just growing so much recently in this field. So even if there's a lot of innovation, there's still a lot of issue, there's still a lot to do in single-cell data analysis. So with this, I conclude the talk, and if you've got any questions. Thank you.
Questions?
So first of all, thank you for answering all my questions. I have one last question. Well, one first last question. Probably grab you afterwards. But my sort of most pressing question right now would be, so we've been talking about cell identities. Mm-hmm. And so my question is, Is that an artificial construct for us to be able to sort of cluster, as you say, the expression profiles that we find? And is there a correspondence with the actual cell types? Because I was thinking perhaps like what we call here bulk RNA-seq. You find that what you see is a sort of average constructs of the different transcript that you get, whereas here I was wondering actually, would not that happen as well? That we are not making a difference in the clusterization, looking at the actual cell boundaries, but rather what we are looking is sort of different average profiles of expression, whereby what you end up having is sort of a computational breakdown of the data that you get that doesn't necessarily correspond to the real cell types. So I was wondering what you think about that.
I think you should use all the data that you have because you don't know the truth. So you look for validation of your analysis. Oh, that's always the case. So you can use databases, you can use any kind. So the identification of markers for the clusters will give an insight of the meaning of the cluster itself. So you will find external information to validate your findings. So in practice, you can have structural validations like, oh, that makes sense, oh, that looks like a good clustering because my metrics are good. But about the meaning, have I identified a type of cell? Question mark. You can be more confident if you find confirmation externally to overlap to your findings. Yeah, so definitely, if you got bulk that tells you something, use it. If you got literature, there's a lot in literature, there's a lot in databases. So you can find, if you identify a set of genes that is specifically expressed in that cell, for example, that you know that is associated to a specific function that is linked to a specific cell, then your confidence in the identification of the cell type is higher. Yeah.
Any more questions? Eugene.
Should we start?
So the exercise I'm getting today.
Just in practice, with so many clustering methods, publishing, say I've got a data set, I've done the pre-processing, next step is to do clustering. What would you suggest? Should I go through all the method, or should I just use the latest one because the latest one always supposed to be the best?
Well, depends.
So what's your opinion?
Depends how lazy you are. No, I'm joking. So you can definitely use the one that if literature tells you those are the best methods, I would use the best methods. But it really depends on your problem. So if you, for example, if you know that there's a specific characteristic of your data set or your problem that a specific methodology is able to pick up more than the best method, because a method is the best based on a criteria, could be not the best for your data. So I will look into the assumption of every methodology and see what's more for your case. But once you select a set, then you can just compare the results. You can compare the results structurally because there's approaches that tells you which one is the best clustering for your network based on the structure of the network. Like, for example, you can use Robin, the package in R, or after that, you also want mostly the meaning, and so all these other validations. So you continue with the downstream analysis to see which clustering makes more sense biologically to do. So I would use more than one. Yeah.
I think in biology, be skeptical, like in general, of what algorithms are producing, because there's so many confounders and bias and all sorts of things going on in the data. So I think people are using multiple algorithms, checking for, any kind of bias or batch effects constantly at every stage is just a case of being skeptical and not assuming something is a true biological signal until there's some validation, I guess.
Yeah, that's true, because there's a lot of confounders, but it could be things that you don't know about the data, so you look at your cluster and they make sense, and then you find out that you divide it out based on gender or based on any other characteristic that was really what you wanted to look at. And so overall, should be more filtering done, actually. Is another question? Behind? Yeah.
Thank you. I was wondering, have you looked at single nucleotide RNA as well? And can you say something single cell versus single nucleotide data?
The answer is very easy. No.
No.
I've not done it, so I don't know. So have you done anything in that direction?
Single nucleus. Yeah.
So my difficulty at the moment is, as I was telling to Magnus, is accessing new data. Because if you work if you're lucky enough to work in a group that is developing a specific, going in a specific direction, it's very interesting also to. My guess is that there would be need to specific development because the data might be different. I don't know, honestly.
But I did see there was a paper about the differences in biases in the single nucleus that was sort of, because it's more nascent RNA in the nucleus, the length of the gene can have a big effect on what you pick up, so that can be different, right?
Like the oligo-DT biases are quite different because you don't have the polyadenylation signal and so on. So, yeah.
It would be very, very interesting just to compare the bias.
Yeah. Okay, I think we probably have to break for lunch. And just to remind people that lunch is upstairs. I'm sure many of you will take it back downstairs and go outside. But let's thank Luisa once more.
Thank you.